This lecture is on the structure of DNA. It should be a review. The key concept of this lecture is that DNA structure is the same in all organisms. DNA is composed of nucleotides. Nucleotides are the Legos that come together to form a DNA molecule. Each nucleotide is made up of 3 different parts: a phosphate group, a deoxyribose sugar, and a nitrogen-containing base. The nitrogen-containing bases can be one of 4 different types: thymine, cytosine, adenine, and guanine. These 4 bases belong to 2 groups: pyrimidines, which are made of a single ring, and these are thymine and cytosine, and purines, which are made of a double ring structure, and adenine and guanine belong to the purines. We use the first letter in each of their names to abbreviate each of these bases. Therefore, T stands for thymine, C stands for cytosine, A stands for adenine, and G stands for guanine. Watson and Crick were the 2 scientists responsible for determining the 3-dimensional structure of DNA by building molecules. Here they are shown in the image to the right. These 2 scientists realized that DNA is a double helix, that it is made up of a sugar phosphate backbone on the outside with bases on the inside. In the image, you can see a model of this 3-dimensional double helix structure that we are referring to. Watson and Crick, their results built on the work of Rosalind Franklin and Erwin Chargaff. Franklin, pictured at the bottom left, produced X-ray images suggesting that DNA was a double helix of an even width. Her X-ray image is shown to the bottom right. Chargaff's rules stated that A, that is, the percentage of adenine, is equal to the percentage of thymine, and cytosine is equal to the percentage of guanine. Nucleotides will always pair the same way. A summary of base pairing rules are as follows. Adenine will always pair with thymine, and cytosine will always pair with guanine. That is to say, a pyrimidine will always pair with a purine. This is why the double helix is of a uniform width. A 2-dimensional view of the double helix shows our backbone. So the backbone of the DNA molecule is connected by covalent bonds, which I am circling right now. The bases, on the other hand, are connected by hydrogen bonds, depicted in this image by dotted lines.